Greetings. My name is Dr. Shanta Messerli, and I'm a scientist in the Cancer Biology and Immunotherapy Group at Stanford Research in Sioux Falls, South Dakota. This study is focused on breast cancer, which is the most common cancer in women worldwide, with a lifetime risk of 1 in 8 developing breast cancer in industrialized countries. Metastasis, the process by which cancer spreads, is responsible for over 90% of cancer-related deaths, including breast cancer. My overall goal is to eliminate mortality associated with metastatic breast cancer. While we talk about breast cancer as if it's one disease, it is in fact heterogeneous and an umbrella of phenotypes, including a range of diseases, each of which has distinct molecular characteristics. One of these types of breast cancer is called triple-negative breast cancer. This form of breast cancer has the worst prognosis, is most likely to progress after chemotherapy, and has the greatest chance of developing distant metastasis. Triple-negative breast cancer has been named after the fact that these tumors are lacking two hormone receptors, including estrogen and progesterone, and are lacking overexpression of the human epidermal growth factor receptor 2, known as HER2. In contrast to other forms of breast cancer, triple-negative breast cancer does not have tumor-specific treatment options, which include targeted cancer therapies directed against these receptors. Since there are few effective treatments specific for triple-negative breast cancer, treatments that reduce metastasis would have a major impact on the survival of patients. One class of drugs that holds this promise are histone deacetylase inhibitors. These inhibitors modulate gene expression by altering chromatin structure, increasing acetylation of histone proteins, making chromatin more accessible to transcription factors, which result in modulation of gene expression. Histone deacetylase inhibitors have the therapeutic potential to reverse aberrant epigenetic changes associated with many cancers, including triple-negative breast cancers. Another potential molecular target for tumor therapy is the histone lysine-specific demethylase 1, LSD1, which plays an important role in carcinogenesis and is an emerging option for treatment of cancers. LSD1 specifically demethylates both lysine 4 and lysine 9 of histone 3, and can act as a co-activator or co-repressor, depending on the context. Here, we demonstrate that the small molecule dual HDAC LSD1 inhibitor 4SC202, also known as domanostat, is cytotoxic and cytostatic in both mouse 41 and human triple-negative breast cancer cell line MB231 in cell culture. The drug reduces cancer cell migration, tumor burden, and metastasis in vivo as compared to vorinostat, which is a broad-spectrum histone deacetylase inhibitor used clinically to treat cutaneous T-cell lymphoma. So in figure 1, we show that epigenetic targets such as class 1 HDACs are upregulated in human triple-negative breast cancers tumors relative to normal tissue, suggesting that these epigenetic markers may serve as an important therapeutic target. In figure 2, we demonstrate that 4SC is both cytotoxic and cytostatic in triple-negative breast cancer, while figure 3 shows that the drug inhibits migration. Figure 4 illustrates that the 4SC significantly reduces the tumor burden compared to a vorinostat treatment. Transcriptome bulk RNA sequencing analysis, which is in figures 5 and 6, from 421 tumors revealed changes in metastatic-related pathways in 4SC-treated tumors, including significant changes to the expression levels of genes implicated in focal adhesion and extracellular matrix receptor interaction pathways. So in summary, the increased expression of epigenetic modulators such as HDAC1 and LSD1 in human triple-negative tumors indicates that these may be important targets for therapeutic intervention. We demonstrate that the dual HDAC1 LSD1 inhibition with 4SC has potent effects against breast cancer cells, including cytotoxicity, inhibition of proliferation, and reduction of metastasis. Bulk RNA sequencing results indicate changes to the expression level of genes implicated in focal adhesion and extracellular matrix receptor interaction pathways. So based on these findings, more preclinical studies need to be performed to investigate the potential beneficial effects of 4SC202 in reducing metastasis of triple-negative breast cancer. you have any comments or questions, please contact me at shanta.messerli@sanfordhealth.org. I would really like to hear from you. Thank you.